Hello, so throughout our videos we have talked about how genetic material gets passed from the one cell to another and how it is contained within the cell and how it affects the inheritance and etc and etc. But we must address the one question from our videos. So if an organism starts from the one cell zygote, it grows into the monstrous size. It consists from the billions and billions of cells. How is it able to do that? It has to some kind replicate itself so that all the genetic material is same within all the cells, other than sacs or germ cells. Well, it happens due to the process called mitosis, which gives us the two identical copies of the cell. Well, until then we have to explain the cell cycle. And cell cycle dictates what cell does and if it passes through the mitotic cycle or it rests and performs its functional duties, or what does it do in general. Well, it starts with the G1 phase, and during this phase it grows in size and performs its functional duties. After that, it arrives at the checkpoint 1. This checkpoint 1 determines if it passes to the G0 phase or resting phase and continues to do its functional duties, or gets a chance to replicate its DNA and passes into the S phase. Well, most of our cells are into G0 phase, but when we are developing and doing embryogenesis, many cells are going through the cell cycle. During the S phase, the DNA gets replicated, so that each chromosome, which consisted from the one chromatid, as we explained in the first video, you can check it out, now consists from the two chromatids. We have to also acknowledge that DNA is not condensed and it is still accessible by some enzymes to maybe activate some genes. After replication finishes, we pass to the G0 phase, where the cell grows even larger, and you arrive at checkpoint 2. This checkpoint 2 decides if the cell is able to pass to the M phase, and it checks for DNA damage and if cell has enough materials to pass into the G0 phase. And if it is able to, it will pass the checkpoint 2 and pass into the M phase, where mitosis happens, and in the middle it also has the checkpoint 3, and we will discuss it later on. And in the end, we get the two identical daughter cells, and each daughter cell can go into the same cell cycle, and that's it. Now let's explain the mitosis. Mitosis consists of the 4 phases: prophase, metaphase, anaphase, and telophase. And we also have the cytokinesis, which is not part of the mitosis and will be explained shortly after. And let's start with explaining the first phase, the mitosis prophase. So, the main guys which play a role here are the centrosomes. Centrosomes are made up from the 2 centrioles, which is arranged in T-shape, and create microtubules to actually connect to the chromosomes and pull individual chromatids, the sister chromatids, apart to the different poles, so that the genetic material can be distributed accordingly in half. The duplicated genetic material, of course. Here we have the nucleus and nucleolus, just the main guys, and here we have the 2 sister chromatids. And during the prophase, many things happen. First of all, the chromosomes get very condensed, so much that it is visible under light microscope. Nucleus and associated structures get destroyed, and the centrosomes move to the opposite poles and create a mitotic spindle. So, these guys over there are mitotic spindle, and at the last stage of prophase, called prometaphase, the process finishes and they move to the opposite poles. After that follows the metaphase. During the metaphase, all the chromosomes are moved and they are migrating to the metaphase plate. It is usually in the middle of the cell, and mitotic spindle also gets larger and larger and tries to connect to the metaphase plate. Here we have the many different kinds of the microtubules, the parts of the mitotic spindle. First are the kinetochore microtubules. They get connected to the central they are connecting to the centromeres of each chromosome, we realize that two kinetochore. They ensure to get connected to the microtubule, and during the next phase, in anaphase, they destroy microtubule in such a fashion that the individual sister chromatids get propelled to the opposite poles. Here we have the astral microtubules. They connect to the opposite side of the cell at the poles, and they actually fixate the poles. And you also have the interpolar microtubules, which stabilize the mitotic spindle as whole by interacting with each other. They are unstable, but they are in large quantities, so ensure to do such a job. And here we also have the third checkpoint, where it checks if all of the chromosomes are connected by two microtubules from opposite sides. And in anaphase, the process starts. The sister chromatids are pulled apart and moved to the opposite ends. And in the telophase, all of the chromatids, now called chromosomes, are moved into the separate parts of the cell. Mitotic spindle dissolves, and all of the structures gets recreated, and the mitosis ends. But as we know, they're still in one cell membrane, the same membrane. In most of the cells, when divided mitotically, they also recreate some of their part of the cell membrane, so they have a different cell membranes, and that's what happens during the cytokinesis. At the contractile ring, it cannot be in middle. It can be also divided into the asymmetrous parts, but here it is here we have the symmetry. The contractile ring pushes the two ends of the cell membrane, and it causes the two cells to separate from each other. As such, we have two individual cells in the end. And in conclusion, the cell cycle and its checkpoints control what and when a cell has to do certain things. Also, it controls if everything is done correctly. And the mitosis is the mechanism that produces two identical daughter cells. Thanks for watching, and see you soon.